So thank you again for joining. I think we'll go ahead and get started. I know people may still be joining over the next few minutes, but I think we'll get started with just the introduction and everything. So let me go ahead and share a presentation here, and then we'll get started. So thank you again for coming. We're joined today by Dr. Jacqueline Stevens for another one of our exciting NIRx webinars. So she'll be talking about measuring brain activity during functional movements and why fNIRS is an optimal neuroimaging tool for rehabilitative neuroscience. And we're joined also by Cami. She's going to be joining us today to help out with everything in the background. I'm Jeremy. I'm the scientific consultant lead at NIRx here in North America. So just a quick introduction to NIRx. So we're a company that's been, for the last couple of decades, striving to support neuroscience research and a lot of different areas, an expanding field with fNIRS neuroimaging technology. We really strive to make sure that we're empowering the field, disseminating knowledge that's gained by many, many labs. And one of the ways that we do that is through webinars like we're doing today. There are a lot of ways to connect with us. I would say, our main goal is to provide education and information about what's going on in the field. So if you have any questions, please don't hesitate to reach out to us via email, all the socials, and there are a lot of resources on YouTube, including things like this webinar that are great to check out. Another way to stay connected with us and up to date on what's going on in the field is through our newsletter. I'll leave this QR code there for just a second, but there's very few of these that go out on an annual basis. They feature a lot of what's going on in research and what are our new technology developments within NIRx. And other exciting news, don't hesitate to sign up for this. There's not a lot of messages, so you won't be spamming your inbox. Just a couple of considerations for the webinar today. Everybody that's joining, you are muted. If you have questions, comments, please feel free to Zoom chat them. So throw them into the chat at any time and we'll be able to review those. We'll have time for questions sort of at the end of Dr. Stevens's presentation. So we'll be checking out those as well as questions that were submitted when people were registering. All this should be available on our webinars website in the next coming days, as well as on YouTube. And if you have questions, any concerns, please reach out to us via email at these addresses. So Jacqueline's joining us today from Colorado State University. She's the director of the SCORE lab in the Health and Exercise Sciences Department. Jacqueline's background is a little bit more focused on occupational therapy. We've worked a lot with Jacqueline in the last few years. Her areas of research are evaluating a lot of different situations with brain injury related to sports concussions, severe forms of traumatic brain injury, more mild cases. She's really been utilizing fNIRS as a tool for a number of years and has a lot of expertise in this area, especially in areas where participants and the recordings are in a mobile situation. So I will go ahead and turn everything over to Jacqueline to start presenting the work that she's been focused on lately.
Thank you. I am going to share my slides here. All right. I think you can see those. Let me know if you cannot. Great. All right. Well, thank you so much for that introduction, and thank you so much for being here today. Like Jeremy said, I am going to share a little bit of information about the studies I've been doing where we're measuring brain activity during functional movements. I do think that fNIRS is an optimal neuroimaging tool for rehab neuroscientists like myself. I consider myself a rehab neuroscientist because I have a background in occupational therapy, which is under the broader umbrella of rehabilitation science. And then I have a background also in cognitive neuroscience. That's where I got my PhD. And I think it's very important for us to understand how the brain works when people are doing things that are very functional and very meaningful. So you'll see that reflected in the protocols that I share with you today. So the protocols I'm going to share today are all movement based, and they all use the Near Sport 2 from NIRx. I'm going to talk a little bit about static and dynamic balance. We have acquired these types of data before and after intervention in adults with acquired brain injury. I'll share another protocol that we've been developing for a long time, which is dual task assessment, where we have motor and cognitive tasks happening independently and then simultaneously. We use these to assess upper and lower body function. I'll say a little bit more about what I mean by that. We've acquired these with athletes with repetitive mild traumatic brain injuries and then never injured controls. And then we have a couple newer treadmill protocols that we've been involved in. These are particularly interesting because, as you'll see, they don't allow for a traditional block design. And so I'll tell you what I mean by that. And we have a couple examples, one where we're using treadmill training and virtual reality for people with moderate and severe traumatic brain injury. And then we have a split belt treadmill training protocol for people with multiple sclerosis. And I'll explain what I mean by each of these because these are very rehab focused terms. I want to make sure that what I'm saying is clear. So again, as Jeremy mentioned, if you have any questions, go ahead and throw them in the chat. Thank you to everyone who submitted questions in advance. Luckily, I'm the kind of person who prepares a talk the day before, so I got those yesterday, and as I was prepping this talk, I was able to embed the questions that I had at the time. So I had about 5 of the 7 questions. So if you don't see your question reflected, I'll try to get to it at the end, or I'll try to weave it into the content that I've already got prepared. As always, this work that I'm presenting represents my work, but also a team of people who support this effort. So I'll share those folks with you towards the end, and then we'll go through questions that I haven't managed to address in the presentation and any new questions that you all have. All right, so I'm going to start with this first protocol of evaluating static and dynamic balance in ABI. Again, ABI is acquired brain injury. So balance impairments are a known consequence. Injury includes traumatic brain injuries like you would experience from sports or a car accident where there's an external force that causes damage to the brain, but it also includes more internal processes like stroke or brain tumors or things like that that affect the brain tissue. And folks across the spectrum of acquired brain injury tend to have some balance impairments. And I work with a yoga researcher who has adapted yoga protocols to support improved balance. And just to show you an example here, this is a subset of 7 people with acquired brain injury. And along my y-axis, you can see the Berg Balance Scale. The Berg Balance Scale is a common assessment of balance that's used in clinical settings. And we can see that if we look at the group averages before yoga, so there's a baseline and pre-yoga. The baseline was we assessed them, and then we did nothing for 8 weeks, and then we assessed them again. So we collapsed those scores together. And then they did 8 weeks of yoga. And so this is their post-yoga score. So you can see as a group, everyone's getting better. This is a statistically significant difference. And then you can also see, because we had a small sample, we were able to plot each individual person, and you can see that their balance actually gets better. So each person's performance from pre to post is plotted here. And just one really quick thing to note is that dashed line across the plots. Any scores below that line indicate that people are at a fall risk. When they score above that line, they're no longer at a fall risk, or they're at a substantially reduced fall risk, which is a clinically meaningful outcome. So we had this information, and we wanted to understand the neurophysiology behind these improvements in balance. We asked the research question in adults with ABI, does yoga or adaptive exercise improve balance or neurophysiology? So we had pretty robustly showed across multiple studies, and I don't have them all mentioned here, that yoga is improving balance. We thought that maybe there was something special about yoga, or maybe it was just the fact that people who were more sedentary were getting out of their homes and doing something. And so we compared it to adaptive exercise, and then we looked at both balance and neurophysiology. We looked at neurophysiology with fNIRS, and we also looked at it with fMRI, but I'm just going to share the fNIRS findings today because that's what you all are here for. But one of the questions that comes up, and this is an important question, is how do we determine what brain areas to analyze? So when we use fMRI, we can get whole brain evaluations and analyses. We can look at functional connectivity across all of the regions of the brain. But of course, with fNIRS, especially portable fNIRS, we're limited to fewer regions of interest. So we select a subset of all the possible regions of interest, and we're really selective about this. We do this all a priori. We set up the cap in advance using our best guess of what regions of interest are going to be most affected or most influential in balance performance. And so here we used a review paper that I have listed here from Surgeon and colleagues. It's a review paper that looks at the structural brain correlates of balance and balance training in humans. And importantly, this paper identified superficial cortical regions that were accessible through fNIRS. So we used this paper to guide our ROI selection, which was the bilateral inferior parietal cortices and the bilateral motor cortices, which includes primary motor cortex or M1 and supplemental motor areas. So that is how we selected those brain areas for this study. We used a block design and a force plate to assess balance and the neurophysiology, again with fNIRS. So as you can see from this diagram, we started with fNIRS cap placement, and then we started recording, and then we started with just a baseline of seated rest. I'm still not sure if we need those data. I'm pretty sure we should probably be getting them in standing. I can speak more about that later, but we wanted just a baseline where they weren't doing any task at all. And then we have them move through our block design where they have their eyes closed on a soft surface, eyes open on a soft surface, eyes closed on a firm surface. So that's just like right on top of the force plate, and eyes open on the firm surface. And this force plate is telling us their movements, their lateral movements, so side to side medial lateral movements, and then anterior posterior movements, so forward and backward movements. And it's giving us that really discrete information about how much they're moving when they're being asked to stay still. And then we're recording with fNIRS to see what brain activity, what task dependent neural recruitment patterns we're seeing during these balance tasks. And again, we do this before and after interventions to see what changes. And then I have questions. I always have questions about what software I use. I swear I'm not employed by NIRx. I use a lot of NIRx products, though. So we do use the NIRx product of Satori. So we've been using Satori for the past, I think, 4 or 5 years to process and analyze our data. I had been using other softwares like Homer, SPM, fNIRS, and I prefer Satori over all of those other options because it is graphic user interface design or GUI based. And it allows me to more quickly analyze and visualize my data. I do use the default pre-processing steps. I'm not going to get into exactly what all of those steps are because they're in page 6 of the publication that is listed at the bottom of the screen here. So we list out every single thing that we do. We do the default pre-processing steps that are available in Satori. This does include short separator regression. We do not have an accelerometer in our cap yet, so we do not regress out those data. That would be important. We're working on getting some funding for that. However, one of the pre-processing steps that we do not use is we don't use the automatic channel removal. We actually process our data twice. We do it with and without automatic channel regression or removal. And we look at the file that the channel removal step generates that tells us which of the fNIRS channels would have been eliminated. And then we look and see if those are having a robust influence on our outcomes. So it's kind of a sneaky way that we're using that information, but we're not like dropping all of the data because the algorithm says to remove it. But we still want to make sure that we don't have a channel unduly influencing our findings. And again, I can say more about that. And then within Satori, we use a GLM to generate beta coefficients for oxygenated hemoglobin, deoxygenated hemoglobin, and we do that per channel, per condition, and per participant. And then we use those values to do within and between group analyses to examine differences. And again, we're staying at the channel level. I'm going to share a little bit in a moment about how we've moved from channels to ROIs, and I think there's some trends in the literature about this. My statistician feels like the channel level analysis is a good option, and doesn't actually think that we need to control for multiple comparisons, because each part of the brain and each channel that we're over is a distinct area. So it's an interesting thing, and I think it's an interesting point of conversation. And I am not wedded to any of the analyses that I've been using. We're just trying to do what's best practice and keep things moving forward to answer clinically relevant questions. If we find out that another approach, maybe an ROI approach or using corrections for multiple comparisons, is better, we will move in that direction. But we're just trying to continue moving forward and answer the research questions that we have. Just as a quick note, this is all I'm going to say about analysis, because we have been using Satori for all of our protocols. So these are the steps that we're using. There, as I mentioned, we do shift between channels and ROIs, and I'll explain what I mean by those ROI analyses later. But this is the only slide that I have talking about analyses, because we're using the same analysis procedures for all of our protocols right now. Okay, so really quickly, in case you're interested, we wanted to know, does yoga and adaptive exercise improve balance? And it does. I'll show you these data really quickly. So what I'm plotting here on the y-axis is the amount of sway. So more sway, so higher along this y-axis indicates poorer balance, whereas lower values indicate better balance. And you can see from pre to post, we have less sway, so we have better balance. And we actually see this in both of our groups. So it doesn't matter if they did yoga or exercise, they both get better. So this is important to us as clinicians. We like yoga. We think yoga is good for folks, but it's great to have two tools in your toolbox rather than just one. So if folks need to improve balance, you could offer them yoga or adaptive exercise, and they both seem to improve balance. There do seem to be a few differences in how these interventions change balance. We don't have enough data to really make strong conclusions about this. It does appear that the different balance tasks also elicited increases and decreases in motor planning regions after intervention. And again, after intervention, there was more brain activity. I'm going through this really, really quickly because we did a webinar on this in December, so I'm plugging my own webinar where we talk about these results in a lot more detail. It's also in the paper that I have mentioned here, but the take-home message here was that there was more brain activity in regions of the brain that automatically respond to balance demands, like the supplemental motor area, and less brain activity in regions that respond to external cueing, like premotor regions. We took this as a possible way to our possible interpretation, again, not too wedded to these interpretations because our small sample size, is that balance may have become more automated after these types of interventions, which is of course a good thing for folks that have balance impairment. All right, so that's the first protocol. Again, I went into a little more detail on analysis here, but I'll keep addressing the selection of ROIs as I move through the other protocols, and I'll tell you how we shifted in our analysis plans with our other protocols. Oh, and just a real quick summary on this one. So we improved balance, as I mentioned. The intervention type didn't matter. We may have detected significant changes in neurophysiology, and this is important because we want to understand how and why interventions are successful, because this adds to the evidence base, gives us more justification as therapists for providing these types of interventions. We do know that there are issues and test-retest fNIRS measurements. Since this study, we have acquired, or since all the studies that I'm going to share with you today, we've acquired an optodigitizer so that we know where our optos are placed. So we're trying to use that to improve our test-retest reliability, but that's a known limitation of the work that I'm sharing today and something we're trying to address in the lab. So we want to make sure that we really are measuring intervention-induced change and not just change because the cap's not in the same place from test to test. And we want to make sure that we can replicate these findings and confirm them with other neuroimaging methods. So our team right now is looking into our resting state fMRI data to see if we see similar brain regions that are different after intervention in our population. We also have questions that we're hoping to answer in future research studies about if these neurophysiological changes persist or if they eventually change brain structure. I don't think in an 8-week study we have the capacity to see change in brain structure, but maybe down the road, maybe if we do longer studies, longer interventions with kind of tune-up sessions, we might be able to see some of those. So these are some ongoing research questions that we have and things we are hoping to explore in the future. Okay. My next protocol that I'm going to share with you is about dual task assessment. And this has been a large component of my work for, gosh, probably like 7 or 8 years now. So, a little bit of background. So, repetitive mild traumatic brain injury. Mild traumatic brain injury is another word for concussion. So, we know lots of people experience concussion, athletes in particular. We also know that even after athletes are cleared to return to play, they tend to have some dual task impairments. So, what I mean by this is that if you go from a single task, so let's say you're just walking, and that's this represented by the triangles, and then you move to a dual task, so say walking and talking at the same time, your performance on both tasks declines a little bit. But what we see in people that have repetitive mild traumatic brain injury, even those that have been cleared to return to play, is they have a much larger dual task cost. So our research over the past few years has been to look at this unknown piece of what's happening with neural attention recruitment. So, are we actually seeing that these dual task costs are attenuated by the amount of neural attention resources that are available and how much people are recruiting? Is there something that's unknown about how the brain is behaving under these different task demands that would help us understand some of the sequelae of mild traumatic brain injury? So our research question here for this protocol is how does repetitive mild traumatic brain injury affect dual task performance and its associated neural recruitment? And again, we used fNIRS for this. And for this study, we've exclusively used fNIRS. We don't have an MRI piece. So again, here we used a block design, and we have spent a lot of time refining this protocol. We're calling it the neuroimaging compatible dual screen now, or the NCDTS, and it's exactly this block design. So again, I've got some resting or sitting at the very beginning, and then I have a single cognitive condition, a single motor condition, and a dual task condition. And as I mentioned at the very start, which I don't expect you to remember, is that there's both a lower extremity and upper extremity. So the lower extremity task is one that's been done in a lot of dual task work where people are walking. So the lower extremity is the lower limbs are walking. It's a gait task. And here we have people stepping over obstacles while they're walking as quickly as possible. And then our single cognitive task is a verbal fluency task. So I give them a letter and I ask them to say as many words as they can that begin with that letter. And then the dual task combines those. So that's our lower extremity task. Our upper extremity task is a little more novel. We don't see it as much in other literature. A lot of people are focusing almost exclusively on the lower extremities. But our upper extremity task was brought about because we know athletes use hand-eye coordination in addition to a lot of lower extremity function for most sport performance. So we wanted to see how hand-eye coordination was affected in dual task paradigms and also the neural underpinnings of that dual task performance. So here, our single motor condition is a throwing and catching task where they take a tennis ball and toss it against the wall and catch it with the opposite hand. So we don't have a hand preference embedded in this. So they're alternating left hand, right hand, left hand, right hand. And then our single cognitive task is a serial subtraction task. So we give them a number and we say subtract backwards by 7 from this number. And then, of course, the dual task is when they combine those together. So we do this in a randomized block design so that we can average brain activity across those different conditions. Here, we are looking at the ROIs of the right lateralized prefrontal cortex and posterior parietal cortex and our bilateral motor cortices. These were generated from pilot fMRI data that I acquired as a postdoc, which was ages ago now. But in those pilot fMRI data, we were seeing hyperconnectivity between motor regions and attention regions, primarily right lateralized attention regions, like the right prefrontal cortex and the right posterior parietal cortex. So we placed our fNIRS optodes and situated our channels over those regions because our fMRI data were suggesting that at rest there was a little bit of odd connectivity, a little bit of odd activity happening here in folks that had mild traumatic brain injury. In the data that I'm about to show you next, we have moved to ROIs rather than channels for analyses. And so really quickly, just the behavioral data first. How does repetitive mild TBI affect dual task performance? So the behavioral performance. It didn't. I mean, it didn't in a group difference. So as you can see here, the red blocks are our repeat SRC. I apologize for you terminology. These were the figures from the paper. So we called it repeat sports-related concussion. So again, sports-related concussion, mild traumatic brain injury are the same thing. But the repeat SRC group, those are our folks that have had multiple head injuries. And then the blue boxes are the controls. And as you can see from these boxes, everybody's performance gets a little bit worse when we go from single task to dual task. So single motor to dual motor, single motor to dual motor, and then the same for the upper extremity. So you can see that everybody's getting worse as we expected, but we don't actually see differences between the control group and the repeat SRC group in the behavioral performance. But what's interesting is that when we look at task-dependent neural recruitment, is that we actually see that for the single motor task, our repetitive concussion group, our repetitive mild TBI group, is recruiting more brain regions to support performance than controls. But when we get to dual task, they recruit fewer. We see a very similar pattern on the upper extremity task. So we found this really, really curious, and we're still trying to wrap our head around exactly what this means. When we were thinking about the single motor, we're like, oh, this makes so much sense. So many people with concussion say it just takes me so much more effort to be able to do these tasks. But then when we get to dual task, they seem to disengage, but we're not seeing dramatic drops in performance. However, we see that most people have resolved dual task performance, but we have heterogeneity in our group in terms of time since injury. So if you know concussion research, people really want to know how long ago their most recent injury was. And we have people who were years from their last injury. The people who were closest to their most recent head injury actually had pretty poor dual task behavioral performance and the most robust differences from controls in their brain activity in both the single and dual motor. So overall, while people have increased brain activity during single task and decreased brain activity during dual task, we do see there's varying time since injury in our cohorts, and that maybe is having an influence on some of these findings. And we don't think that dual task performance is resolving for all individuals with repetitive mild TBI. So we're working towards getting additional data with people closer to injury to really understand who's recovering, who's not recovering, so we can kind of understand that heterogeneity. And we're working on a grant proposal right now to see if multimodal evaluation can provide insight into why some people appear to have residual impairments and dual task performance and other outcomes, whereas other people seem to recover completely. So we're really trying to use fNIRS and our other measures to tease apart some of these heterogeneous findings that we see in this population. Okay, I promise I'm going to stick more to the protocols in these next couple of slides. All right, so the next protocol that we're doing is very interesting, and it was a challenge for me because, as I mentioned at the beginning, we have had to move away a bit from our block design approach as we move into some treadmill training protocols. So the population I'm going to talk about here is moderate and severe TBI. So these are both types of ABI. And as I told you earlier for protocol 1, you have balance impairments in ABI. We also have gait impairments, so that shouldn't be terribly surprising. If your balance is off, your walking or gait movements are also affected. There's been a good amount of research to show that in clinical settings, treadmill training protocols are really helpful at improving performance. And we wanted to understand what the neural underpinnings of that improved performance are in a traumatic brain injury population. So, this is what the treadmill training protocol looks like. And as you can see here, it's a small picture, but you can see here this person does have the fNIRS cap on. So, we call this VR. This is what the hospital, this is Craig Hospital, which is a TBI Model Systems location. They are using this treadmill, and if you can see in the image here, there is the option for body weight support, so the person can be in a harness that is actually supporting a bit of their body weight. And then they're walking on the treadmill, and when we say VR, it's this game that they see on the screen. So we can either have this screen on and they're engaging with the game, or off. And they're calling this virtual reality. It's not fully immersive virtual reality like you think when you have like a VR headset on, but this is how this is described. So it's kind of like a gamified treadmill training protocol. And we were really curious to see what the neural underpinnings of improved performance, when we see improved performance, are in TBI. We started with a feasibility study, and I'm going to show you a little bit of data from that feasibility study. Here, we selected our ROIs based on motor regions that are involved in walking. And because there's this gamified piece, we also wanted to look at bilateral prefrontal cortices to see what type of attention allocation was being used. And here's where things get interesting. So we have this protocol where they have to do a continuous task. That is the intervention. And so we can't have them starting and stopping and starting and stopping like we do with a block design that we've done for balance in our dual task assessments. So what we decided to do here is get some brain activity from early on in the task, in the middle of the task, and then later on in the task. And then we had the treadmill training either with the treadmill with no screen on, no VR on, and then they would do the treadmill with the VR on, or we'd counterbalance the order where they'd start with a VR and then do the treadmill afterwards. We counterbalance this between all of our participants. And everybody was at a preferred walking speed. Both of the tasks were 12 minutes in duration. So we use this to see, first of all, is it feasible to acquire these kinds of data so that we can answer that research question of what are the neural underpinnings of improved performance? We had feasibility benchmarks, and we also had this exploratory or bonus research question, which I'll get to in a moment. But the feasibility benchmarks that we used for this study were, is it safe? Can we do this safely? Can we put this backpack on, even though we know it weighs about 2 pounds or 900 grams? I think it's 900 grams, but a very small amount. Does it influence their balance? These are folks that have balance impairments. If we add something to their back, is that going to throw things off? Are we going to increase the risk of falls? Is the paradigm acceptable? So folks with brain injury have a much higher proclivity for having headaches. We're going to put a cap on their head and make them have the cap on their head for a long time. Is this acceptable, or is it super uncomfortable, and is it something that a lot of people aren't going to be able to tolerate? We wanted to know if we could acquire high quality data, and then if we could analyze those data. And spoiler, this paper is under review, but the answer to all of our feasibility benchmark questions is yes. So this worked the way that we had hoped it to. We had no instances of falls or any other adverse events. We had a acceptability questionnaire that we used with folks, that we adopted from another study, that's also included in the paper. And the majority of folks said, yes, this is fine for me to wear. And we were able to get high quality data from 7 of 8 participants. And we were able to analyze this data to answer our exploratory question was, is brain activity different with and without VR? So I looked at those last 3 epochs. So towards the end of the task, I wanted to compare with VR on versus VR off. And in that contrast, where we look at VR on, we see a lot more brain activity. Again, this is such a small sample size, we can't really interpret too much from these data, but it might suggest that the VR on is much more cognitively engaging and taxing for people, which is an important insight for rehab practitioners to have, because we would assume that the VR task would elicit more brain activity, and some people need that, but some people need a lower grade activity in their rehab protocol. As a rehab protocol. So it's important for us to be able to validate how much more effortful a task is. And that's kind of one of the advantages that fNIRS is giving us here. Okay, I'm moving to my last protocol, and this one is not entirely mine. This is a grad student's dissertation work that I'm on his committee, and I get to share his work with you today. I think it's super cool. I hope you do as well. It's another treadmill protocol. In this case, it's for people with multiple sclerosis. Again, we have gait and balance impairments in this population, and they negatively impact quality of life, especially because gait and balance impairments contribute to falls. A main predictor of falls is walking asymmetry. So when you have asymmetrical walking patterns, I'm going to show you what that looks like in just a moment. And this is a common consequence of stroke, Parkinson's disease, multiple sclerosis, spinal cord injury, etc. So this affects lots of different groups of people. So this work could have an impact on MS and beyond. So this video should show you what walking asymmetry looks like. So if you can see here, this person, he's walking and he's leading with his left leg, and his right leg is kind of dragging along. So this is a really good example of walking asymmetry, and we see a lot of this in MS. So there's a split belt treadmill training protocol that's used to try to improve this. So it starts with overground walking, just as you saw, where you have a lead leg. The person has a lead leg that's working a lot harder than the other leg, and it's their less affected limb. And then we use this to determine what this person's preferred walking speed is. Then we move to a tied belt, which is a traditional treadmill. This is, if you go to the gym and hop on a treadmill, it would work like this. So it's called tied belt, or just your regular treadmill. And you can see how the person walks on this. And we also can assess their preferred walking speed on the treadmill. And then we do a split belt, which is a rehab intervention designed to change and adapt gait mechanisms. And in this split belt, the affected limb is forced to walk at a faster speed, a significantly faster speed than the less affected limb. So if somebody is left leg dominant, or their left leg appears to be stronger or less affected, they speed up the treadmill track on the right side, and the left side actually slows down quite a bit. So that's what this looks like. And then you can see after this protocol how the person is walking. And now you can compare it to their original gait patterns. So you can visually see the amount of improvement that this treadmill training protocol elicits. And so this is Andy Hagen's work. One of Andy's research questions for his dissertation is, what are the neural underpinnings of this improved performance? And so we have been working together to apply fNIRS in this study. So Andy has really been interested in the ROI approach. And so he has all of his regions of interest are organized by ROI. And so all of the channels that he has included in his montage are labeled here for you to see based on ROI. So we've got the supplemental motor area in green, premotor cortex in orange, primary motor cortex in red, primary somatosensory cortex in blue, superior parietal lobule and inferior parietal lobule in purple and pink, respectively. Again, these ROIs came from the literature, from fMRI studies. We are using these in analyses to understand the neural underpinnings of baseline performance and then improved performance in folks with MS. Oh, sorry, one more. And we've got the superior middle temporal gyrus in yellow. In addition to this, and I'm not going to go into this in a lot of detail, Andy has introduced transcutaneous electric neural stimulation, or TENS, into his protocol, which you can see on the furthest right photo there. That is known to improve some of this gait asymmetry in MS, and he has used that in his protocol to see how that changes brain activity as well. It's a fun video here. You see the brain activity during the task. All right, and then just very briefly, this is his entire protocol. So he has folks with, PWMS is persons with multiple sclerosis and healthy controls. They come in and they get this TENS stimulation first. before they get it. They have no tens. Then they have a 4-week washout period, and then he has tens off or tens on for their second visit. So that's his counterbalance protocol for looking at how this treadmill training and with and without neural stimulation is affecting brain activity. And this is the important part for you all who are interested in fNIRS protocols. So I showed you what the split belt treadmill training protocol was. And just like the other treadmill training, we can't start and stop it. We can't put it in a randomized block design like we would hope to, like we would normally do in other types of studies. So we have these continuous walking blocks, what he's calling early adapt blocks, middle adapt blocks, late adapt blocks, and then that de-adaptation period where they're no longer on the split belt. So de-adapt, early, middle, and late de-adapt blocks. And so Andy has been looking at the brain activity within all of these different blocks with tens on and tens off. He's got some really, really cool findings, and I promised him I wouldn't share them yet because I didn't want to steal his thunder. And his results are coming soon. He does have a paper under review with all of these data, and he'll be defending his dissertation this semester. But this is some really cool work for you all to look out for, and his last name there is at the bottom in the under review publication that we hope to have in press soon. All right, I'm going to get to all of your questions, but before I do that, I just wanted to acknowledge that, again, all of this work is done by tons and tons of people. We have financial support, we have mentors, we have collaborators, we have trainees, and all of these people do really, really amazing work with fNIRS data collection, with the interventions, with supporting people. When you work with folks that have neurological conditions, they often forget their appointments and they get lost when they're coming to campuses because college campuses are confusing. And so we've got people that are picking people up and helping them navigate campus and calling them and emailing them and sending them multiple reminders. So this cannot be done with a small team, and it does take a village to do this kind of work. And I'm so appreciative to everybody who's on the team and supporting this. Okay. So I'm going to get to your questions, and I think you've been encouraged to drop those in the chat. I also had a couple questions that I didn't get to in the presentation. One of those questions was, what is the advantages of NIRx devices for measurement with movement? When I acquired my NIRx device, it had the most amount of head coverage for a portable system, which is why I selected it. I'm not on the market for a new fNIRS device, so I don't know if there's other companies that have portable devices with more head coverage, but so far I have had a really great experience with my NIRS Sport 2 device from things break, and literally Jeremy has brought me a new device when mine needed to be repaired. And Satori has been really helpful for rapid and consistent analyses. And so I've been really very happy with the device that I have and the support that I've received with it. And again, I'm not sure that there's a competitor that has more head coverage, although I could be mistaken. What solutions am I using for long-term data storage and sharing? So far, our NIRS data are not nearly as big as our MRI or EEG data, so I've been able to use them just to store them on just university network drives, which are, of course, secure and restricted. And as far as sharing, I'm actually working on a data management and sharing plan right now as part of a grant I'm writing, and so I'm trying to figure out the best way to share NIRS data. I do think the .NIRS and .snarf files that are generated from the NIRS Sport 2 are really common NIRS files and can be used across a lot of different analysis softwares and platforms. So I think if we put these data into a repository to support multi-lab group analyses of shared data, I think just sharing those raw files is going to be pretty useful for most people. Okay, you've heard me talk for a long time. I apologize, I've gone a little longer than planned, but I am happy to take any additional questions that I have now.
Awesome. Thank you, Jacqueline. We do have a couple of questions that have come up in the chat. I can read those to you and you can comment if you'd like.
Yeah.
So the first one is from Fabio. He says, congrats for the presentation. My question is about the baseline signal of the fNIRS. You acquired 60 seconds before the standing task. Did you acquire any other baseline periods between the standing task And how did you use the baseline signal to analyze the data? So this is back in one of the first protocols that you mentioned.
Yeah, so we acquired all those baseline data and we have not used them. So we have been working with the NIRx analysis experts to make sure that we're analyzing our data properly. And in these block designs, because we have the task on and task off, we are able to actually calculate the baseline from the task off periods. And that's actually what we're using in our contrast when we are using the contrast. Because we have different groups and different conditions, we often use those different conditions, like dual task versus single task, as our contrast, rather than using it against a rest period or baseline period.
Perfect. So Matcha asks, thank you for the presentation. My question, besides looking at the hemodynamic responses, changes in HBO and HBR and beta values, which other kind of measures would you advise to look at, report, something like peak amplitude or time to peak, or any of these sorts of measures, something you've looked at?
We have exclusively been looking at beta values. And so that, again, has been a recommendation that we've seen in the literature and recommended by NIRx. There's a great YouTube video on what beta values are and how they're useful. And so we have not been asked to report those. We do report both HBO and HBR, but we're reporting beta values on both of those in our studies. And so far, these align with some of the best practices that have been published. I think it's a USL paper that talks about best practices. So we align with those when we're reporting our data, and we haven't had any pushback from reviewers about reporting additional measures. People tend to be pretty happy with beta values.
Great. Sarah asks, I want to ask where we can learn fNIRS analysis from the beginning. Do you have any suggestions?
Um, Well, yes. So I did go to a Ted Huppert workshop on fNIRS data analysis. Um, it was helpful and complicated. I think some folks that do a lot of this analysis and are really refining analyses, and this is true across the board for neuroimaging, it gets pretty esoteric in the descriptions, and they'll talk about the different processing parameters and how one is different and what it does to the signal. And while I think those are really, really essential and important conversations, I'm not a methodologist. I'm not an engineer, and I am trying to use the information that's out there and the publications that seem to really convince me of the methods that are being used. And again, in collaboration with other people who have lots of expertise in this, to pick the best processing parameters and analysis parameters for my study to answer a clinically based research question, because that's really where my expertise lies. And I don't want to get into much of the weeds of the methodology. This isn't really answering the question, but it's, it's part of the answer. So, I mean, I do think these workshops can be really helpful. I think Satori manuals are really helpful. I am self-taught in a lot of my analysis steps and understanding, which I think is just kind of an evolution of becoming, an associate professor, is you just have to learn these things, and you learn them at workshops. But I also, I have offered to and continue to work with other labs and teams on questions that they have with this, and I just kind of walk them through from the beginning to the end of what we're doing. And I think attending these webinars and reading papers and seeing what people are doing, you'll start to understand those basic analysis steps.
Great. Yeah, I'll just quickly plug, we're hosting a workshop, NIRx, in Orlando next month, which will include a lot of analysis training, and Ted Huppert will be there to sort of provide that. And our goal is to have other workshops, like Jacqueline mentions, that are focused on analysis, at least in the US, even this year. So agreed that there's a lot to learn from the papers and publications, but basically I think you covered a lot of the resources. All right, another question: how to differentiate between motor and cognitive, the signals from dual task? You mentioned a lot of the comparisons between motor performance and cognitive and motor dual tasks, but maybe more about how do you separate out the different things with the performance with cognitive and dual task?
Yeah, so we can kind of look at the single task performance, and this is where we'll use the calculated baseline from Satori to say, okay, what's the brain activity when we do just like single motor to baseline, and we can kind of see which regions have increases in HBO, for example. And then when we do the dual task, where we know we've added on the cognitive, it's like, what is the additional? And then we can do that contrast where we subtract out the single motor from the dual. And so we can say, what is the added brain activity from adding the single cognitive task? And we can do the same thing for the single. So single cognitive task versus baseline. We can also look at all of these metrics in the context of the behavioral performance and whether the behavioral performance got worse, like it does for the majority of people, stay the same, or in the rare instances, some people actually get better when they dual task. We don't really like advertising this because we don't want people texting and driving, because that's like the typical dual task that we tell people, don't do this. Things get worse when you try to do these things. But we do see those exceptions where some people actually have a dual task facilitation where they get better. So we can look at, again, the single tasks compared to baseline, the dual versus single, and then look at all this in the context of behavioral performance to see if we have atypical behavioral performance to help us understand exactly what the neural underpinnings are for each of the components of the dual task protocol.
Perfect. So another question, did you also calculate the neural efficiency for cognitive load?
I'm not sure if I fully understand the question. Can I get a clarification on that?
Yeah, Elham, if you wanted to write any
Hi.
more about that,
Hi.
go ahead.
So the neural efficiency, which is based on the amount of oxygenation and the behavioral measurements. So there is a formula for that. So I'm not sure if you're familiar with that, but I know that some people do that, also calculate the neural efficiency, because they say that it uh, fNIRS measurements only mean something when it is calculated or it is considered in the context of behavior. So
Yeah, so we are calculating things like dual task effects. So that gives us that behavioral component of again what I was talking about of dual task interference versus dual task facilitation. So we do have that context of the behavioral performance. We are looking at relationships between brain activity, so like just simple correlations between channels that are more active during one of these conditions. And then we look at that in the context of behavior. And we have seen pretty robust relationships there. We haven't done a specific neural efficiency calculation. I'd be curious to see what that formula looks like. But we have looked at other types of relationships between the neural activity and the behavioral performance. So we are absolutely considering the brain activity in the context of behavior, which I think is the recommendation that you're mentioning. But yeah, I'd be very curious to see what that is if you wouldn't mind sharing that paper and that formula.
sure, I can do that. I'm not sure if I have your email address, but I can certainly do that.
Yeah, I can. Let me just drop it in the chat real quick. Probably the fastest way for you to get it. There you go.
Perfect. I received it. Thank you.
Yeah, thank you.
So Jacqueline, there's another question here. How can we account for non-brain signals such as changes in scalp blood flow in paradigms when NIRs is worn during treadmill movement rather than
Oh,
getting balance?
sorry, that's such a great question because I forgot to mention that we have short separators in our caps. So the short separators are measuring that scalp perfusion, and in our analysis, we have the short separator regression included so that we can regress that noise from the signal. And then as I think I mentioned, I can't remember, I'm in the middle of writing a grant right now too, so my brain power is power is limited. But we are working on getting an accelerometer to add into our cap so that we can quantify the amount of head movement as well and regress that from our signal.
Perfect. Okay, well, I'll give space to throw any other questions into the chat. Cami, you don't have to tell me where we're at with time, but
I think we can go ahead and close the webinar.
Sounds good. So yeah, thanks again, Jacqueline, for your presentation, for sharing all of this valuable information. I know there are a lot of groups out there looking to do similar types of movement related recordings, so I'm sure it's very helpful for many, and will be. And thanks everyone else for joining, and have a good one, and we'll see you next webinar.
Thank you.
Bye, everyone. Thank you.